Hi everyone, Arsalan Arif here with Endpoints News, and this is part 2 of a 3-part webinar series on powering clinical research with AI. Today's focus is on faster, more efficient trials, novel trial designs using digital twins, sponsored by Unlearn AI. My guests today are Charles Fisher, Unlearn AI's founder and CEO, and Jonathan Walsh, Unlearn AI's founder and head of data science. They've got an approximately 40-minute presentation, and we're going to follow up that with about 20 minutes of Q&A. During part 1 of this series, we had a ton of questions. So we're going to make every effort to get to them all. So just hit that Q&A button at the bottom of your screen to submit yours, and do that as early as possible, please. Charles is going to start us off, and then John's going to pick up the second half of the presentation. So now, without further ado, I give you Charles Fisher.
Thank you very much, Arslan. Let's see if I can share my slides. Excellent. So to follow up on our first presentation, I'll just give a quick recap that we at Unlearn AI have developed some technologies for creating what we call digital twins. And a digital twin is a type of simulation, a computer simulation, that helps us to ask what would likely happen to a particular patient in a clinical trial if that patient were to receive a placebo. And the question that we're going to cover today is how can we use that type of information, that computer simulation, to create novel kinds of clinical trial designs that are going to be faster and more efficient than traditional randomized controlled trials. And again, just to recap a little bit from the first presentation, and this is something that everyone in drug development is very familiar with, that clinical trials are incredibly slow and risky and expensive. This is often encapsulated in this thing that people call Eroom's law, which is the reverse of Moore's law, that over time, drug development as a rule is getting much, much more difficult and more expensive. In contrast to many other things in society that are getting faster and cheaper over time. And there's a few different statistics that we can look at. So one is that only 10% of drugs that actually enter into clinical trials end up as approved therapies coming out the other side. And 80%, so the vast majority of clinical trials, experience delays in enrolling subjects for the trial. And in fact, 20% of clinical trials fail to ever meet enrollment targets. So if you look at sort of this entire process, there's a clear bottleneck. And that bottleneck is finding subjects who fit the inclusion and exclusion criteria of the trial and are able and willing to participate. And this bottleneck makes it much more likely for clinical trials to be slow or to not succeed. And this is on top of a general environment of increasing competition and shorter periods of exclusivity, where second-to-market drugs now typically come on the market only 6 months after the first-to-market drug. And that's a big change from how things were 20 years ago. And these things are particularly acute. There are problems that are across many indications of drug development, from rare diseases to cardiovascular trials, but they're particularly acute in neurodegenerative diseases. And Alzheimer's is a really prototypical example. In a typical Alzheimer's phase 3 pivotal clinical trial, there'll be more than 800 patients. The trial typically takes more than 4 years and costs more than $200 million. And despite all of these patient volunteers, all of their time, all of the time of the researchers and the investment from pharmaceutical companies to run these trials, there hasn't been a new approved treatment for Alzheimer's disease in over 15 years. And of course, we'll find out more information on that soon. But this really highlights an unmet need for a way to do things differently, to develop drugs better and faster for these types of complex disorders like Alzheimer's. And we mentioned before that there's one of these bottlenecks, and that bottleneck is finding patients. And this leads to slow clinical trials that take a very long time. And this is a problem that we can solve today. And the idea here is very simple. Can we take, say, an Alzheimer's trial that would typically have 1,000 patients and use new technology, use existing data to reduce the number of patients that we would need to run that trial while still keeping all of the statistical properties of the trial that we care about? And if we could do that, reduce the size of the trial, reduce the number of subjects that we need, then these problems with patient enrollment that cause clinical trials to be very slow would be alleviated. And we could have much more efficient, much faster, and less costly drug development in Alzheimer's disease. And when we think about this, there are a number of different clinical trial characteristics that we care about. And we can develop different types of clinical trial designs to balance these different characteristics depending on where we are in the drug development life cycle. So one of these things that I've talked about a lot is speed, making sure that the trial is fast and efficient. But that's actually not good enough, especially if you think about for pivotal trials. There, it's also important that the results of that trial are robust and reliable. And that means that we also need to think about how we can run analyses that are going to be robust to what we call confounders. These are unknown unknowns, patient characteristics that may affect the treatment outcome and cause us to attribute a difference between a control and a treated population to a drug when it was really caused by something else. And then in addition, there is controlling the type 1 error rate, something that's very important in a lot of clinical trials. And what that means is, what's the probability that we would accidentally approve a drug that really in reality doesn't work? And so that's something that regulators in particular are often interested in guaranteeing that type 1 error rate is controlled at a low probability. And what we're going to do today is go over a handful of example analyses of a clinical trial to illustrate what the advantages and disadvantages are of different novel clinical trial designs that incorporate digital twins to help get more efficient and better estimates of treatment effects. And we're going to do this by taking a previously completed clinical trial in Alzheimer's disease, this trial by Quinn et al. From 2010, that had approximately 400 patients in a 3-to-2 randomization ratio. So 3 patients received the active experimental treatment in this trial for every 2 patients that received the placebo. And we're going to reanalyze this trial under different hypothetical scenarios. And before we get into those scenarios, I do want to mention, just recall that in the previous first installment of this webinar, we talked very briefly about 2 different ways that we can think about evaluating the evidence in the trial. One of these ways is called the frequentist approach. And that approach is really defined by this idea of controlling the type 1 error rate. So we say, if the drug doesn't work, I want to make sure that there is a very low probability that I would call a trial positive. And I can fix what that is, what I'm willing to accept, and then I can develop a statistical analysis plan based on that constraint. There's another type of trial way of evaluating evidence called Bayesian methods. And those methods incorporate other background information that we have from, say, previously completed clinical trials to try to improve statistical power above and beyond what you'd have in a frequentist approach. So that's going to be important, and we'll get to more detail about that later. So we're going to simulate 3 different hypothetical trials. The first one is a single-arm trial, the type of thing that you might run in an early phase proof of concept study. So here we had in the original trial 238 patients who received the active treatment. So we'll simulate a scenario where the same number, all of those patients who were enrolled into the active treatment, they would receive the treatment in this hypothetical scenario. But 0 patients will receive a placebo. And instead of having actual patients receiving placebo, we will take the baseline data from each patient in the treatment arm, create a digital twin, and then use that intelligent control arm composed of all of those digital twins as an external comparator. In a second hypothetical trial design, this is a type of randomized controlled trial that we will analyze in a frequentist approach so that it's acceptable to use for an early phase or even a pivotal clinical trial, because it will control the type 1 error rate. And in this trial, there are only 193 patients receiving treatment and 144 patients randomized to placebo. So this is a trial with a smaller enrollment, but the same statistical power as the original study. And finally, we'll talk about another example of a Bayesian clinical trial design that has a 4-to-1 randomization ratio and uses the intelligent control arm and information about how accurate those digital twins are at predicting placebo outcomes collected from previously completed clinical trials to reduce the number of patients in the trial that need to be randomized to placebo. So this trial has only 48 subjects who are actually receiving a placebo. And as a result is a significantly smaller clinical trial design, again, keeping the same statistical power as the original study. And so with that, I'm going to turn over the presentation to my colleague, Jon, who will dive into more detail about these hypothetical clinical trials.
Thanks, Charles. I'm excited to tell you about how the role digital twins can play in these designs. I think I need to take control of screen sharing. Can you, I can't share until, thanks. Okay. So the design of clinical trials can be informed by many factors. In early stage studies, speed can really be critical. Fast proof of concept studies mean that effective therapy can be identified more quickly, and pivotal trials can start sooner. At the same time, we still need to be able to draw sound conclusions from these trials. For these cases, we want to utilize designs that minimize the number of subjects, but still provide decision-making power. So single-arm trials can be really useful in this case. In a single-arm trial, all subjects are given the experimental treatment and no subjects are given a control. This allows for quick studies, but we need something reliable to compare against to determine if the treatment is effective. Digital twins can be the comparison. Digital twins in an intelligent control arm are an external control that can tell us how effective the treatment is. While single-arm studies can never fully control type 1 error or be robust against confounders, digital twins can be validated in other studies and utilized in these cases. So why in particular are digital twins useful here? Well, the digital twin predicts the likely outcome of a subject if she were to receive a placebo. To see how that's valuable, let's discuss a patient's role in a clinical trial. When a patient enters a trial, her baseline data is collected, providing measurements of her clinical state at the start of the trial. A digital twin is created from her baseline data. This information, taken before the subject receives any treatment, is used to predict her disease progression if she was receiving a placebo. The patient then continues through the follow-up period, accumulating data that provides a comprehensive picture of her disease progression. If this patient's in a single-arm trial, then she will be receiving treatment, and we want to use this data to determine how effective it is. We want to know how much better she's doing than if she never received the treatment. And this is where digital twins enter. In particular, the digital twin provides a clinical record as if the patient was receiving a placebo and gives us something to compare the patient data against. In single-arm studies, it's really that simple. Using the subject's data under treatment, we take the difference between their outcome and the outcomes predicted by their digital twins, which are modeling progression under placebo, to estimate the treatment effect. Okay, so let's look at our example trial to show how digital twins can serve as an external control arm. If you look at the data from the study, then the treatment arm and control arm outcomes look similar across the time for both ADAS-Cog11 and CDR sum of boxes, which are the primary endpoints of the study. There's some natural variability, but overall this indicates the treatment's not effective. When we replace the control arm with digital twins with the treated subjects, we see a similar comparison. The observed treated and digital twin outcomes are consistent with similar rates and total amounts of progression. We can use results like these to increase the confidence in the ability to use digital twins to accelerate early phase studies in the fastest possible way. So I've talked about primary outcomes, ADAS-Cog11 and CDR sum of boxes, but there are other outcomes in a clinical trial, not just primary endpoints, but secondary endpoints and safety endpoints as well. There's a lot more to digital twins too. They're rich longitudinal data that enable a broad set of analyses and trials, even safety. So on the right, I've shown an example digital twin record from the previous webinar that shows endpoints for ADAS-Cog, CDR, MMSE, which is a common screening tool, clinical variables like vital signs or laboratory tests, as well as background information for subjects. We're only going to focus on primary endpoints here, but I want to show this as an example of the breadth that digital twins provide. Okay, so let's move on from the single arm scenario to a very different type of study. Pivotal trials are an example of a case where control of type 1 error and robustness to confounders is absolutely paramount. Speed can be a benefit, but these trials frequently are going to use conservative approaches to ensure regulatory success. So how can digital twins play a role here? Well, they're created for all subjects in the study, not just for treated subjects. This ensures robustness to confounders, relying on the benefits of randomization to average out confounding effects between arms. And let's talk more about how the role digital twins will play in these tightly controlled studies. So we can utilize the framework of covariate adjustment, which is a common tool, in a really novel and powerful way. By adjusting for the predicted control outcome that each patient is provided for by the digital twin, in fact, the largest gains in power or increase in confidence of any covariate is coming from the digital twin's predicted outcome. And this can be done without compromising the type 1 error rate. So this approach is really powerful, and let's discuss really in detail how it works. When we analyze the outcome of a study, we focus on measuring the difference between the treated subject's outcomes and the control subject's outcomes. We can think about the statistical model on the left, that we have some observed outcome that we're measuring for all subjects. And there's a treatment indicator W that tells us just 0 or 1 and tells us if the subject is in a control arm or a treatment arm. So for a subject in a control arm, we're measuring some control outcome, and we can estimate the average control outcome beta naught. For subjects in the treatment arm, we want to measure the difference between their outcomes and the control subject's outcomes, and we call this the treatment effect, or beta W. So we're going to focus on beta W here and come back to beta naught shortly. The challenge is that in each arm, the outcome that we're measuring can be highly variable. This variability can make it very difficult to identify significant treatment effects. So in the example on the right, we're charting on the vertical lines observed outcomes for patients in each arm. In the placebo arm, we're showing in red circles example outcomes of subjects, and we're showing the effect of a treatment in blue diamonds on the right. The outcomes in these two groups overlap heavily, and it's really hard to tell if the treated subjects actually progress more slowly, meaning if they fare better in this case than control subjects, the treatment's effective. We'd like to explain this variability somehow and help us see the effect of the treatment more clearly and detect significant effects more easily. So let's plot this change in outcome against the predicted outcome from the digital twins for all subjects. What this does is it separates subjects that don't have much progression from those that do, and it more clearly differentiates the two arms, the effect of the treatment. Even though there's a lot of variability across each arm, when you compare subjects with similar predicted outcomes, there's much less variability, and the treatment effect is much clearer. We're measuring the same treatment effect as the case where we did not adjust for these outcomes, but we can lower this uncertainty significantly and make it easier to identify effective therapies. So in a statistical model, these digital twin outcomes are included as an adjustment term. Mathematically, what this does is it allows digital twins to explain the variability in the observed outcomes. Since we're estimating the relationship between the observed outcomes and the digital twin outcomes, in our example, we see parallel lines for that relationship in the control and treatment groups. The bands are denoting some measure of uncertainty where these lines are. The distance between these lines is the treatment effect, and we can see it's clearly much more significant. So this is really the advantage of this approach. It allows for digital twins to explain variability in treatment and control groups and decrease the uncertainty and measure treatment effects, making it easier to identify effective treatments. So covariate adjustment isn't new, and the FDA has recently issued draft guidance that defines how covariate adjustment should be utilized in the analysis of clinical trials. Our approach fits squarely into this guidance, and we're really excited about the potential that digital twins bring to pivotal studies. If we turn to our example trial, let's see how digital twins will impact the analysis and design of this study. So we've taken the original study with a 3:2 randomization ratio and designed a 4:3 randomized version with nearly 20% fewer subjects. In this smaller study, we were adding digital twins for every subject in both arms into an intelligent control arm. And we're using the predicted outcomes with the methodology we just discussed. So the result is very encouraging. The design with digital twins has a result equivalent to the value and confidence of the larger original study. The observed average treatment effect and its uncertainty are consistent with those in the original study, with approximately 20% fewer subjects in the trial. This isn't just an accident. The frequentist method that we're using mathematically guarantees these gains that are dependent on the quality of the digital twins and the ability to model the subject outcomes. So finally, let's discuss a case that combines the best of all worlds. When speed, robustness, and type 1 error control are all important, we can employ Bayesian analyses. Bayesian methods allow us to utilize digital twins more powerfully while still allowing for a degree of type 1 error rate control and robustness to confounders. So first, let's discuss how Bayesian methods differ from frequentist ones in terms of their approach to analysis. This is really where the control outcome becomes really important. While frequentist analyses only use the data in a study to estimate treatment effects, Bayesian methods bring in prior knowledge, some kind of information from the world, to make estimates more precise. And really, why shouldn't we try to add this prior information? We know so much about Alzheimer's disease progression, and we've run so many trials that we ought to have learned something useful to understand these by now. So we're not really completely reinventing the wheel here. The statistical model that you're seeing is the same as in the frequentist case. The measures of the control outcomes, treatment effects, and the digital twins' ability to explain variability are all present. But what we're going to do is we're going to put some notion of prior information on the control outcome and the digital twin outcome. So these are the things that we really have access to in a study. And what we've learned tends to be, from historical data, tends to be quite powerful. We can use prior data to estimate the control outcome and how well the digital twin outcome models that. This will help make trials much smaller. One thing we usually don't have is a prior on how effective the treatment is. We may have a guess or a hope, but without really being well informed by data, we run the risk of biasing the result of the study. So instead, we focus on the outcome we know a lot about, which is controls. Let's see how this is going to impact our example study. As in the frequentist case, we're going to take the 3:2 randomized study and design a study that includes digital twins. But now it's going to be randomized 4:1, a reduction of nearly 40% of the number of subjects. And as before, we're creating digital twins for every single subject in the study and putting them into an intelligent control arm. We'll analyze the study with the Bayesian methods that we've defined before and utilize a prior for the control and digital twin outcomes. So these results really are stunning. The credible interval and posterior probability are consistent with a treatment that has no effect as expected from the original trial. The design, including digital twins, has many fewer subjects. In fact, if you add these up, you'll notice that it's only got a few more subjects than the single arm case, but here we've run a randomized control trial with a large randomization ratio. So really, this is truly the best of all worlds. So in case you'd like to learn more about our approaches, you can read papers on our website about our machine learning methods and Alzheimer's disease modeling approaches. And then you should look at, for publications that we have in preparation upcoming soon. So I'd like to recap on the three use cases that we've discussed. In a single arm trial, which is really appropriate for early phase studies, speed is paramount, and we want to still control confounders and type 1 error as best as we can. In the frequentist case, for early phase or really pivotal studies, the robustness to confounders and controlling for type 1 error is essential, and speed becomes secondary. We can use digital twins to make trials as efficient as possible while still maintaining the degree of control that we need. And then in the Bayesian case, we want to prioritize all of these things and weigh them by utilizing prior information about studies to make informed decisions efficiently. So with that, I'd like to hand it back to Charles to wrap up.
Thanks, Jon. And so just to kind of, again, reiterate and recap a couple of things that are really important. So the first thing is that, as Jon showed, we can create significant reductions in the number of subjects who are required for a clinical trial, while still keeping all of the statistical properties that we want. We can have a randomized controlled trial that's robust to confounders and that has control of the type 1 error rate. That is way more efficient than a standard just one-to-one randomized controlled trial. By incorporating these predictions of what would likely happen to a patient if they were to receive a placebo, that makes that adding that information about each individual patient is really a powerful method for making these clinical trials much better and much more efficient. And so the other aspect that I think is important to mention is we only presented here analyses of, say, just the primary or key primary or secondary endpoints of these trials, things for Alzheimer's disease that are related to cognitive decline. So ADAS-Cog or CDR sum of boxes, these measures of cognition. But digital twins are rich clinical records. We're talking about novel clinical trial designs that decrease the number of subjects by a lot, 40 or 50% in some cases. And that means that we need to have predictions for these placebo outcomes for every single patient characteristic that you would want to analyze in that trial, so that you're not only are you keeping power on your primary and secondary endpoints, but you're keeping power on all of your other analyses that you would want to conduct as part of a clinical trial. So it is a comprehensive way of running clinical trials with a smaller number of subjects, controlling for statistical properties, and doing that across all of the different analyses of the study. And so that's something that, yeah, we're really excited to be able to tell you more about today. I'm sorry. There is one more thing that we are going to tell you about today. And yes, I did always want to say that. So all of the examples that we talked about so far are prospective clinical trial designs. They are novel ways of running a clinical trial with reduced requirements for enrollment. But digital twins and intelligent control arms can also be added onto ongoing clinical trials because they only take the baseline data from a patient before that patient receives their first treatment in the trial. And we can do whatever we want with that baseline information, as long as the trial's still blinded, then we can do whatever we want with that information and still not bias the outcome of the trial. So let's think about it, something that's happened, been a big problem in clinical trials, really across the disease spectrum, but in areas like Alzheimer's that affect elderly populations has been particularly acute, right? So let's imagine that you plan your clinical trial. You want to have 1,000 patients for your Alzheimer's clinical trial. And then we've seen incredibly high rates of dropout and or low enrollment because patients are not able to participate or to continue to participate in a trial because of basically, in this case, the COVID-19 pandemic. So what happens is instead of having 1,000 patients, you might only have 500 patients. And that decreases the statistical power and makes it much more likely for your clinical trial to fail, even if you actually have an effective drug. So let's think about a way that we could use our simulations to look at this problem. We started with this same clinical trial from 2010 that had about 400 subjects randomized 3-2. And then we can simulate the effect of this external factor, this unforeseen external factor that leads to high rates of dropout. And we'll just randomly drop out 50% of the subjects across the two different arms. And then we can say, well, let's add in an intelligent control arm by taking the baseline data of each one of these individual subjects who's still in the trial and creating a digital twin, regardless of whether or not that subject was randomized to active or control. So again, these are randomized controlled trials, as were some of the prospective designs, still randomized, but incorporate this prediction of placebo outcomes into the analysis using this covariate adjustment method that Jon described. So what impact do these types of things have on a trial? So the original study was designed to have an 80% power for a particular desired effect size for the change in ADAS-Cog 11 and the change in CDR sum of boxes over the course of the trial. So we want to be able to have a high confidence that we can detect sufficiently big effect size on these two key endpoints. But after you delete 50% of the subjects in this simulation, those probabilities are reduced to about 64%. So now you have a significant reduction in your ability to identify these therapies if they're really effective. And by adding an intelligent control arm to this ongoing clinical trial, you can recover the statistical power of that trial back to 80%. So this is a way for an ongoing, already running clinical trial that you can very easily incorporate digital twins to mitigate risk and recover power for these types of unforeseen circumstances that cause low enrollment or high dropouts. And again, this particular method that we describe here is actually a frequentist approach that provides strict control of the type 1 error rate. It is mathematically guaranteed as long as the study is still blinded. So this fits squarely within current regulatory draft guidance about the use of covariate adjustment methods in clinical trials. So this is something that provides a tremendous benefit without any risk to causing unreliable results in the trial. So reliability with efficiency, you can actually get both by leveraging these types of digital twins in your trials. So to recap, then, we've actually described 4 different novel clinical trial designs. So there were the 3 prospective clinical trial designs, a single arm trial, which is very, very fast, reduces the number of patients as much as possible. But as Jon said, we can't guarantee that that type of clinical trial is going to be robust to confounders or that it's going to control the type 1 error rate. We can't guarantee it. There could be differences between the patients that we use to train the model that creates digital twins and the actual patients in the trial. But we can incorporate digital twins onto or into randomized controlled trials. And we can do that in a way that keeps all of the benefits of a randomized controlled trial while providing also benefits from the digital twin. And that is that you can get much faster clinical trials, but still be robust to confounders and still control for type 1 error rate. And not only can you do that prospectively to design faster clinical trials, you can do it in ongoing clinical trials after they've already started to recover statistical power caused by unforeseen circumstances. And I've seen the Q&A. It looks like we have a lot of really good questions, so I'm looking forward to that. Just would like to say, if you want to learn more about these methods, as John said, we have a number of scientific publications on our website. Please check those out. We also are very happy to have in-depth discussions. So you can reach out to us at partnerships@unlearn.ai. And also, I know that I've seen already a few questions in the Q&A related to regulatory, and we might cover those a little bit today, but we are going to have a third webinar on December 2nd, which will be a panel discussion focused entirely on the issue of the regulatory landscape for the use of these digital twins in clinical trials. So we will get to that in depth in just a few weeks. So again, thank you very much. And Arsalan, really excited to have the chat and take some questions.
All right, let's do it. And thank you for that. You're right. We have a ton of questions. We have over 100 live viewers right now. Lots of great questions. So thank you for that. Thank you, John and Charles, for that presentation. So we're gonna try something new. I'm gonna put up the PowerPoint here. So we've got the questions up here. I've taken several from the Q&A. I've grouped as many as I possibly could during your guys' discussion. So let's get to it. I'm gonna read this first question. Maybe this is for you, Charles. Or John, how do you generate synthetic digital twins data based on prior data from clinical trials or observational studies for a new drug with a novel mechanism of action and unknown efficacy and safety profiles? In other words, why would we expect the model that generates digital twins to generalize to a completely novel drug trial?
This is a really good question. Also, I got to see who was asking these questions, and I know who asked this question. So it's really important to think about the clinical trial as being a comparison of two potential outcomes. So this is something that we discussed in the first webinar. You have one outcome, which is what would likely happen to this patient if they receive the new active treatment. And you have the second outcome, which is what would likely happen if they received the placebo or some other existing treatment, right? In one potential outcome, what happens if the patient receives a new treatment? We do not say anything about that potential outcome at all, because the question is right on point. We don't have any data on the new treatment, particularly if it's a phase 2 trial. I mean, there's probably no data on that treatment at all. So there's no way that we could try to use a machine learning method to predict something about which we have no data. So what we are proposing trial designs where patients are real people, are enrolled in the trial, given the active treatment, and you observe the outcome. But the potential outcome of the patients in the control condition involves patients receiving placebos and standard of care. And we have a lot of data on patients receiving placebo and standard of care, both from the large number of previously completed clinical trials in Alzheimer's disease, but there's also just potentially patients from registries or the EHR who are receiving standard of care. All of that information can go into inform predictions of that potential outcome. So the key thing here is that we are making predictions only for the placebo outcome, which is the part of the trial that we can get data on, not on dealing with novel mechanisms of action or anything like that of these drugs. We don't predict that. That's observed in the experiment. We only predict the placebo outcomes.
Okay. All right. Let's move to the next question here that I've selected. Okay. I've grouped 3 of these together. I'm going to read them all out and you guys take them as it comes. Is placebo effect simulated for digital twins, or are they based on normal patient population not affected by anything, including awareness they are in a clinical trial? So one would expect single arm open label patients to perform better than active patients in a placebo controlled study. Do you have any suggestions for minimizing that effect? And lastly, I'll read the number of possible combinations with co-medications, including doses, genotypes, comorbidities, tau and amyloid load far exceeds the number of available patients. Where do you get the placebo data for training your network? And how generalizable is this approach for a completely novel trial in a new population?
All three really good questions. And again, things that if people haven't gone back to the first webinar is up on the website, and a lot of these things are covered there. And so you can still go back and get a lot more detailed information about some of these things. So the data that we use to train these models, by and large, come from the placebo control arms of previously run clinical trials, although not entirely. There is also some observational data, but mostly placebo control arms. So these were patients who knew they were participating in a clinical trial, who had a possibility of being put onto an active treatment. So the data themselves incorporate the placebo effect that you would expect to observe in a clinical trial like that. And we have approximately, so it's not one clinical trial that we are using here as our training set. We've integrated data from 21, John, 21? 21 studies, 21 different studies with around 7,000 patients. And so those 7,000 patients, we do include some, co-medications, particularly things that are used, symptomatic treatments for Alzheimer's, ApoE genotype, some comorbidity information and biomarkers in here as well. So we have a pretty large data set that's been collected across sort of a diverse population because it's from sort of not one place, but many different studies that have been integrated together. John, do you have anything to add about that?
No, that's great. And I think the goal is to be able to model a diverse population across the disease spectrum in Alzheimer's disease or any indication that we're working in. I comment that for completely novel trials in new populations, I mean, that becomes a real challenge because, as Charles said, you need data on the control population to be able to make confident predictions. So the vast majority of trials in Alzheimer's disease are run in similar patient populations with similar designs, similar data collected. That really helps us expand, when we use these data sets, expand the breadth of populations that we can model.
Very good.
I would add one additional thing onto that point. So we talked about 3 different types of trial designs, right? So the single arm trial, if the model were terrible, if it were terrible, it's not, it's really good, but if it were terrible, then it would mess that trial up real bad, okay? But when we're talking about the statistical analysis plan that incorporates the predictions from the digital twins, particularly in a frequentist framework, we can mathematically guarantee that even if the model were terrible, and again, it's not good, but if it were terrible, it would not mess that trial up, right? So it's not just the property of how good the model is, it's a property of the way we perform statistical analyses to incorporate the predictions of the model into the trial. So even if there were a huge difference, let's imagine that we have a new clinical trial that's using a primary endpoint, which has a brand new type of cognitive test that has never been measured in any patient ever before. If you incorporate the digital twins in the frequentist framework, you can still do that, and you can still guarantee that you'll get increases in power from adding the digital twins, and that you will not increase the type 1 error rate. It's a property of the statistical analysis plan that's true, and that's really important thing to keep in mind.
Okay, well then let me push you on this a little bit as a follow-up. Can the single dose trial example be applied to dose escalation or adaptive trial designs? If so, how?
Yes. This, although, so there's a few different things, right? Right now, we've really focused on creating a handful handful of concrete, well-defined statistical analysis plans. And the goal there is that we people can review these plans, and so when we can present evidence and a lot of analyses that really clearly demonstrate the value and the advantages and disadvantages of each plan. Because we want to be able to have evidence that particular plans will work. Can we apply or develop other kinds of plans to more special scenarios, like an adaptive trial? We could, yes. I mean, digital twins could be used there, but we currently have been much more focused on creating these well-defined analysis plans and evidence to support their use.
Very good. All right, let's move to the next question here. This is another placebo effect question here. So, and maybe it's one that was in the previous webinar. And I just also want to remind all the viewers for this placebo effect stuff and a lot of the material that Charles covered in the first webinar. If you open the Q&A section to see the answered questions, we've already had the link to the previous webinar there, so you could watch it right after this and see that presentation. But let me just read this, and Charles, get your reaction. Is the placebo effect simulated for digital twins? Or do we already read this one? Let's move on to the next one then. Okay, are digital twin synthetic data created using statistical methods only, or do you also use some first principle simulation model that simulates toxicity or tumor development? And another safety question, how do you compensate with digital twins that reduce the acquisition of data on drug safety? If one reduces the size of the treatment and control arms, one gives up gathering safety information.
Yes. So starting with the first question, the methods that we use are a particular type of sort of artificial intelligence model. I'll even say what it is, although we use a kind of model called a latent conditional restricted Boltzmann machine. Not that we need to get into detail about what that is, but it's a kind of sort of deep learning style model that makes longitudinal predictions, and that's a key thing. So we're predicting probabilistically, so likely outcomes over time for patients with this kind of sort of neural network model. It's a purely data-driven approach. So we're not putting in any kind of information about how we think the disease will progress or mechanism or anything like that. It's purely data-driven. What that means is that we have to make sure that we have a large enough data set, incorporate data from many sources, and spend an incredible amount of effort. The majority of the effort internally is cleaning and integrating the data rather than doing the machine learning. And on the second question, if you look at the actual example of the digital twin that John showed earlier, it's not just, say, predicting ADAS-Cog11. It's not just predicting CDR-Sum. So not just these things related to the efficacy of the drug, but we are also predicting, say, values of all of the important labs over time. So you have comparator information for things related to some safety. So you're looking at things like liver or kidney function or things like that over time, where I actually are simulating that with the digital twins. We also have some information in these digital twins for Alzheimer's on adverse events again, for the placebo arm population. So this is a really important point, that if all you're doing is predicting the primary endpoint of the trial, this question is absolutely on point, then you would, if you would lose all of the power for everything else, and everything else is still important. So that's why the methods that we use actually output very rich SDTM formatted clinical records that cover all of the patient characteristics that you care about for your trial. So it's not just predicting the primary endpoint, predicts all those other things. So you should be able to still perform these kinds of analyses.
All right, very good. We have just under 10 minutes left, so I'm going to try to fly through these because there are tons of questions in the queue here. As you can see, okay, here's a statistic for simulations. How are the allocation ratios? How are they decided?
John, do you want to take this one?
I'd be happy to comment on this one. So actually goes back to the last question as well. The goal is to provide randomization ratios that preserve subjects in the treatment arm. and reduce the number of control subjects. These randomization ratios that we chose, 4:3 and 4:1, were based on our assessment of the ability of digital twins to model outcomes on historical data sets that we have access to in populations similar to the trials that we're targeting. So what this means is that we have this allocation ratio, the randomization ratio that's predefined, and provides a design where that number of subjects that would remain that are in the treatment arm would be approximately the same as the number of subjects that you would find in the treatment arm in a 1-to-1 randomized trial without digital twins. So really the goal is to preserve patients on treatment and reduce the number of control subjects.
Right, moving along. In your opinion, are there disease states or therapeutic areas that are most conducive to using digital twins at this time? What recommendations would you have for rare and orphan diseases?
So this goes back also to an earlier question about these being data-driven approaches. Because they're data-driven approaches, we really can apply them across any indication, as long as there is data to learn from. And what we've kind of been focusing on initially are areas with high unmet need. So areas where drug development has been difficult, where clinical trials are long and expensive and require lots of patients. Of course, rare diseases is another area of unmet need, maybe for different reasons, that there's so few patients in many cases for those areas. We are interested in doing some work in rare diseases. The key factor there is just making sure that there's a sufficient amount of natural history data for the disease that we would be able to train one of our models. There isn't like a fixed size of how many patients we need to train a model. And because of the way that our models work, as they're longitudinal models that predict changes over time, the size of a data set is something like the number of patients times the length of follow-up. So for rare disease, even if you maybe don't have a very, very large patient population, if you add a sufficiently long period of follow-up in your natural history study, you may still be able to take this kind of approach.
Okay. Let me follow up this question with this question here, which just came in. How long does it take to develop a digital twin in other indications, say an acute disease where the course of disease is fairly well known?
So all of this comes down to a question of how much data there is and how easy it is to clean. So the actual training that we do, we put it on a supercomputer and it kind of trains. And that's not necessarily the longest amount of time, the machine learning part. What does take a long time is if you're integrating data from a lot of sources and there's a lot of variability in how different studies were run, what endpoints were used, how things were coded, then we have to fix all that. We have to go in and map everything to a common kind of data model. And that is hard. So it depends really on the state of the data that are available.
Okay, next question here. Okay, well, again, this is a regulatory question over here, and I'm just going to make one more plug for part 3 that's coming up in our 3-part series, which is just going to deal on regulatory issues over here. So unless you have anything more to add to that, Charles, I'm just going to implore all the audience to sign up for number 3 on this one, and we'll get into all of those topics.
Yeah, just very briefly. So we, Jon mentioned that there is regulatory guidance or draft regulatory guidance from FDA and EMA on the kinds of statistical analysis plans that we're proposing that suggest that those plans would fall underneath the existing draft guidance. We have also ourselves had individual meetings with FDA to talk about this approach, and Will can provide more discussion around that in the 3rd webinar.
All right, very good. We're just ending on the closing minutes of this webinar, so let's get through a couple more questions here, and let me know if you've already answered some of these, Charles or John. How would digital twins assess safety and tolerability, account for discontinuations due to safety for drugs with different MOAs?
Yeah, I would say again that the goal is to provide digital twins is to provide a data set that's comprehensive for these types of assessments that you want to perform during a study for the control population. So drugs with different mechanisms of action or drugs with different safety profiles that want to be assessed, our goal is to provide historical data via the digital twins that can be used to carry out those analyses.
Okay. All right. Let's try to get a couple more questions in here before we have to wrap up. Is there a reason the data for digital twins was consistently skewed down from the control group?
Yeah, I can comment on this. I think probably referring to the single arm case in the example trial we showed. So in that study that you can, this is the single arm case. None of the points that were shown were statistically significantly different. So there's no, if you analyze the trial at any time point, the treatment effect would never be statistically significant, except for possibility if you analyze the actual study at 12 months, in which case, if you go back and look, you would say, well, maybe the ADAS-Cog is getting better and better for the treated subjects and still getting worse for the control subjects, the relative difference.
All right.
Yeah, at the same time, what the digital twins are bringing in is you can think of as a historical view of the expected amount of progression for that patient population. And while it still remains consistent, it is just a bit lower than what you see during the study.
Okay. All right. And this next question, I believe you have already addressed this one because, Charles, you were talking about the possible minimal sample size of prior data. But do you have anything to add to this question here?
Again, there's no such thing necessarily as a minimum sample size. It depends on how complex of a model that you want. So if you want to have a model that simulates many different things, you need a bigger sample size. If you want to have a model that simulates a smaller number of things, you need a smaller sample size. So it depends on the use case. And so there isn't necessarily a hard and fast rule, but we're happy to discuss particular applications with people if they reach out to us.
Okay. All right. Okay, I try to group these together. I think you've addressed elements of these questions also. We've got about 2 minutes left and I'd like to get a little bit more of your insights here. How can digital twins be reliably generated for rare disorders? Number of patients is limited. What solutions exist to this issue? How do you generate synthetic digital twins for data for conditions where symptoms vary over time? These are essentially some questions that you have already answered in the past. Anything to add on these?
Well, I think we kind of covered the first one about having longitudinal data. The second one, I think actually Alzheimer's disease, what we're showing here neatly falls into this category, right? You have a large number of symptoms there. You have these cognitive tests that are measuring all these different dimensions of cognition over time. We're simulating each dimension. So we're not simulating just an endpoint. We simulate every symptom itself. And what that means is it doesn't, whatever endpoint combination you want to create some composite score, you can use any of them. I think that this actually is exactly where you want AI. You want AI for these complex things where you have many symptoms evolving in a complicated way over time. If it were simple, we would be able to use simpler methods.
Okay. All right. Let's see if I got one more. No, we've already, so those are the questions that we have for today. And now we're reaching up on the end of our webinar. So I'm going to implore the entire audience again, if there's any questions we didn't get to over here, I will endeavor to get to you. But please, number 3 in the series is going to be about regulatory. And number 1 in the series, you can go to click on the Q&A button, look at the answered links over there and click on that and check out Charles's whole presentation for that. Again, I want to thank both of you, Jon Walsh and Charles Fisher from Unlearn AI, for this presentation today. And thanks to all 150 plus of you who attended today and gave your questions. For Endpoints News, I'm Arslan Arif, and I'll see you next time.